So we check in the onion and then cell of tobacco leaf cell. And then we observe by confocal microscope. Actually look like very easy, but practice is very difficult. And our results show that this is control. So GFP fusion protein, this protein localized in the cytosol and then nuclear. Where is our location, our protein location in the cell? So this is using N-terminus, so PGW5TIL1 GFP. In onion, our protein localized in the plasma membrane very clearly. But this is in the tobacco. In case in tobacco, our protein also localized not only in plasma membrane, but also localized in the reticulate structure, and then also in here we can see in the nuclear and then plastic. And also same data when we check SLTIL2 in tomato. This result show also that the control, that protein localized in cytosol and in the nuclear. But after, in the GFP fusion transition, so that the data, protein clearly localized in the plasma membrane in onion. But in the tobacco, the protein localized in the reticulate structure and also in plasma membrane. So, and then we check the several journal that Abu et al. In 2014 said that this protein in Populus eupratica in this plant is clearly localized in plasma membrane. But TIL in Arabidopsis by Gross et al. In 2015 localized in plasma membrane and reticulate structure. And our data show that this protein not only localized, so we not follow in this group and this group. In our data show that this our protein localized also in the plastic and nuclear. So that means this result show that TIL protein not only localized in plasma membrane, but reticulate punctuative structure and also localized in plastic and nuclear. What that means? That means this ribocalin function the protect organella membrane in plasma membrane. So we try to making transgenic tomato using this protein. So we transform TIL protein into microtome tomato, and then this construct we check the ploidy by flow cytometry. This is the vector. This is the method how to transformation protocol. So Professor Azura from Tsukuba University was successful making transgenic tomato using some gene to the microtome. So we followed that the protocol. And our result went to making the transgenic tomato. So we got that 29% or 36 line from 700 line this candidate transgenic. So 26 line is diploid. This is microtome transgenic. And then after that, we want to check the immunoblood analysis. So why we making the transgenic tomato? Because when we study about the function of protein, we should be compare the wild type and with the transgenic tomato. So we can understand more on the function of the protein. And the next experiment, so we want to know the confirmation the proteome data, and then the comparing this element is protein region and the promoter region, and then using this, consider this reason how to making the transgenic tomato. We want to check again, make sure that where is the subcellular localization of this protein using transgenic tomato. And then we want to know also protein-protein interaction. Because sometimes some protein first localized in plasma membrane, and then after binding protein, so interaction with other protein, that protein migrate to reticulate structure, sometimes like this. And then we also want to know the post-translational modification. So sometimes some audience ask to me that what the benefit of this research. So we have many problems with abiotic stress in the world. So many problems, plant disease and others. So we want to, using transgenic tomato, compare with wild type and compare with normal plant, we can check, and then so we will treatment using the abiotic stress like high temperature, low temperature and others, and then salt tolerance, and then zero tolerance and others, also methylfulogen or chemical effect to the plant. After then, we will check the RNA isolation and then by RT-PCR. So, and then we will check the phenotype. If our gene is clearly have function to this, so I think we can help and support some science to the world, so applied science, that in any country maybe have problem about the abiotic stress. So I think enough my presentation, so thank you for your attention.
Thank you for the talk, Mr. about the lipocalin protein in tomato. Now the session is open for the discussion. Please.
So why the plyD level is changing in your transgenic plants?
Sorry, one more.
Why plyD level is changing in your transgenic plants? Why?
Yeah, ploidy level. Yes, thank you. So when we check the ploidy level using flow cytometry, so some plants actually we use the diploid plant, but some plants is changed to the tetraploid. So the mechanism actually I cannot explain detail, but sometimes some genes when how to say, when we make construct with other factor, some genes sometimes change the ploidy to tetraploid or others, but I'm not sure about this because our data show that 50% from candidate transgenic, when we check the flow cytometry, that 50% only diploid, but 50% tetraploid or others, like that.
Any other question?
And one more question. You said that it localizes to plasma membrane.
Yes.
So is it a membrane protein? Did you check with any database or something?
Yes.
Is it a transporter or something?
Yes.
It has transmembrane domains.
Okay. So in our picture show that maybe we can, yeah, and here we can clear when we know in the cell, this is tobacco leaf cell in the onion epidermal cell. This is clearly in plasma membrane. We also check by bioinformatic tools, like when we check CHL chloroplast, we use chloro-P and target P, for example, like this, and also result same. So that means our data also using the software and using the particle bombardment is same result.
So very short question.
Okay. The till 1 and till 2,
Yes.
are they the isomer or is a completely different protein?
Actually, sequence of very
And
identical.
how many, I mean, the percentage of identity in nucleotide level between
the-
these two? Yeah.
In the nuclear,
Yeah. No, in nucleotide sequence, the till 1 and till 2, how many percentage there is identical?
identical. Actually, TIL1 and TIL2 in the every plant a little bit different, but this is very similar actually. So identical at least 99, more than 90% maybe.
Okay. Thank you again. Join me to thank him again. And now let me invite Dr. Kurab Bashir. He's from Riken, Yokohama. He's going to talk in strategies to develop crop plants tolerant to different abiotic stresses. Please.
Okay, thank you very much for the organizers for providing me a chance to share my interest. I will not go into much experimental details. I will rather take this time to introduce my research area, because if I go into experimental details, so maybe it will be very confusing for understanding other people. Okay, so I basically work on plant molecular biology, abiotic stress response, mineral nutrition, biofortification. So we eat every day. And we often also say that we are what we eat. But how many times we wonder what we eat and what kind of food we get from when we eat? We have basically energy. Everyone knows calories, how many calories you have. But then comes, we also take vitamins. And some vitamins are synthesized in body and some from food. And we also take minerals. And remember, human bodies or animal bodies cannot synthesize these minerals. You have always acquired these minerals from outside. And this is two proportional types. One is that humans, they need these minerals. From where they get? They get from food. From where this food comes? Majority from animal sources and plant sources. And again, animal and plant sources also need these minerals for their healthy growth cycles. So where to acquire them? Animals graze plants, and for plants, to acquire these minerals from soil. So, and it's not easy. And if you look at the systems, you will understand how intelligent plants are. We often look very down to plants very down. And before going into details, this is just kind of few risk factors for human health in developing countries. Hunger, everyone knows. Unsafe sex, I will not go into details. Unsafe water and sanitation. So this is also two ways where one is we drink water directly, of course, so we can take care of it. We will go and buy mineral water. But again, plants also need water for growing. If water is not healthy, so plant doesn't have the choice to buy mineral water, they have to survive in this water. And when they use this water, they accumulate a lot of undesirable things which ultimately enter into the food chain. And we end up eating this without knowing that. This problem is particularly severe in poor countries where food safety standards are not implied. You are growing food for yourself and eating without knowing what does it contain. And then come zinc deficiency and iron deficiency. This is a major deficiency in humans and plants. And approximately 30% of the human population is suffering from this. Just imagine, 30%, 2 billion people. Most of them even don't know, especially which are not educated, which are mostly dependent on vegetarian food. They don't know. 30% of the world population, 2 billion people. And the problem is so severe when we talk about hunger. It's only, we say, only poor people suffer from hunger. But when we talk about these kind of deficiencies, there's no discrimination between poor and rich countries. This is data from some good countries, and you see that even from Japan, you see that around 40% of the population suffer from iron deficiency. This problem is particularly severe for females and children because their demand is high, and because females go through menstrual cycles, so they excrete a lot of blood which contain iron, they need more iron. And this problem is very complicated, so there are some very nice stuff from previously about the function of the proteins. So we talk about my duty is, my part is the characterize the function of each protein one by one which are involved in this mineral transport. But problem is that they sometimes are not very selective. The similar example is that you go to market to buy something, but they are very similar looking products, not exactly same, and you end up buying with the wrong product because it looks very similar. So plants sometimes face this. They want to transport iron, but they end up transporting cadmium, maybe manganese, zinc, copper. And we are making model kind of things that understanding what functions, but our understanding is still not very good. You see that this is Lego blocks, not Lego blocks, it's some other kind of blocks. So we can make a model which looks like original, but it's not original. So this is our current state of understanding. And how does these minerals and other things interact with each other? This is a very simple example. We grow plants on 3 kinds of media, which are exactly similar, but iron concentration is different. So it's not surprising that among these 3 plants, we measured iron concentration in leaves and roots. It's not surprising because there is no iron, there is less iron. in roots and shoots. If we supply moderate iron, so it seems good enough. If we supply a bit toxic excess, so they accumulate a lot more in shoots. It's very easy to understand, but you will be surprised to know that when there is no iron, zinc increases, copper increases. Here you see copper increases, and again if there is excess iron, copper again increases. Manganese again increases. So this is the poor selection by the proteins. So we basically characterize these proteins, and when I say that plants are very intelligent, so you will see that they recruit, just like recruiting. You hire a person, I am thirsty, I don't want to move, I request brother, please give me a glass of water. No. I request you, please bring a glass of water for me, and you bring it. So plants do use this strategy. So what they exactly do, they synthesize some chemicals. So when we talk about the soil, there's a lot of minerals. But when we say a lot of minerals, it doesn't mean that they are available. They are in chemical forms which are not soluble in water. They are there, and plants can only take things which in contact with them by root. If they reach contact with root surface, plant can take in. But if it is even 1 centimeter or millimeter away from roots, if it is not soluble, it will never come in contact with plant root. How to make it soluble? Do something. So one way is that they reduce it. I will not go into detail. I will mainly focus on. So plants secrete some chemicals and excrete into the rhizosphere, in the water. These are in the water. Now when they come in contact with some metal, they bind it. When they bind it, it becomes soluble. When it becomes soluble, the chances that it will come in contact with the root, with the water flow, increases, and plants takes it. So they excrete, bind to iron. This iron becomes soluble and is taken up by the plants. So we basically work on how these chelators are synthesized. So this is basically we know the chemical pathway here, and we cloned here. This was basically quite old data, 2006 JBC, and then recently we published in Plant Journal. I was a co-author, and we are revising for Journal of Experimental Botany. This was the first report in Plant Kingdom. And we use a lot of techniques. I will not go into details. We can see that when these kind of proteins, when there is enough iron, plant are wise enough, there is almost no expression. There is very low expression in the regions which are involved in long distance transport. But when there is no iron, the expression is the increased synthesis. Expression increased color means the increased synthesis. So they need more recruitment. And we have involved in a lot of other strategies. This was one of the examples. So we used just to acquire function. There's one technique introducing, from cDNA we make cRNA. The cRNA is injected into oocytes, which makes protein, and then we add the substrate which we want to check if this protein is really transporting this protein or not. So we can confirm the function of this. And we also propose that beside these previously known chelators, plants are also release some phenolic compounds like protocaffeic acid, and these phenolic compounds also help in solubilizing plants, which are later reduced to more soluble form and then taken up by the plants. So have been also involved in the subcellular transport of this, how plant transport these metals from cytoplasm to mitochondria or others. This is very pure molecular biology techniques. We have published this in Nature Communications, Plant Signaling Behavior, RISE, and very recently in Viganing and Bashir et al. So this is also, it was first report in Plant Kingdom. So we use many kind of techniques like screening tDNA library for iron deficiency symptoms. So we grew plants in the presence of iron, but some plants cannot utilize this iron. This iron is in available form, so they show symptoms of iron deficiency. People also look yellow, slightly pale if there is iron deficiency, the color, and plants look more pale. So, and then we characterize, I will again not go into details, but just some molecular mechanisms. If plants cannot transport iron, this is the protein name, to mitochondria, plants actually die. So there's no question, it's so essential. But even if this activity is slightly reduced, in this case 30% activity reduced, they still have 70% of its original activity. But still it shows a very significant phenotype. And what happens actually, the plants accumulates in cytoplasm, iron, sorry. And this iron becomes toxic. And it could be a toxin actually. So to avoid this toxicity, they have used upregulate many genes they store in vacuoles. Sometimes they divert this to chloroplast and they use these different mechanisms to, and we understand it reasonably well. And these are also some actual applications. When we disturb the transport, we can actually make plants which accumulate more metals in edible parts. Like in case of rice, rice is a very poor source of minerals, especially white rice. Very poor source of minerals. And we can increase these minerals in rice so that especially this is a very good idea for rich, poor people who cannot eat meat every day, who mainly depend on rice for their daily activities. So we can also stain iron. This is iron. You can see if you make it in this kind of mutant, the iron is less. In this kind of mutant, there's more iron. So you can actually regulate this. And this has been actually applied for different kind of things. For example, in Japan, the problem of cadmium accumulation in rice, it was permanently solved. So they will release a variety. Even you grow in the presence of cadmium, it will not going to absorb any cadmium. And we can increase, also extract cadmium and other toxic metals from soil, so soil will be more clean for future use. And we can also use, make plants which accumulate more minerals for healthy diet. Thank you very much.
Thank you, Dr. Kurabashir, for the great talk. Now the session is open for the discussion. If you have any, please.
I just want to ask about the plant. Usually the plant needs like a good soil and good water, right? Just to like have a good mineral, right? So about the water, like what kind of water? Like is it pure water? Is it like, what is the source of the water? What kind of water we need?
So actually we have very less control. It depends on local conditions. So what kind of, if it is, you have a river nearby, of course farmer will use this river water. But if there is no river or something, they can sometimes extract groundwater.
Okay.
But sometimes even it's difficult to extract groundwater, it's too costly, so they just depend on rain. So, but for these minerals which I am talking about, iron and something, some minerals are not present in soil, are poor. But these minerals like iron, manganese, they are always present. Iron is too much in excess in soils. Approximately 6% of the soil is iron. It is always there, but for some reasons plants could not use it, so they have to develop some strategies or something.
Okay, please.
So far we know that in animal body, iron is transported by some plasma protein like transferrin or ferritin. So actually I'm a background of animal science. So is there any such type of carrying protein, iron carrying protein in plant? If so, so in case of iron deficiency, what is the phenomena of this type of carrier protein which carries iron?
So hepcidin-like things have not been discovered in plant yet, but ferritin do exist. But unlike animal ferritins, which animal ferritin is in cytoplasm, but plants do contain ferritin only in chloroplast and in a very little quantity in mitochondria. So system is to some extent similar, but some extent different.
So in this context, what is the carrier of iron in plant?
So we have local synthetic chelators which are not present in animal system, like deoxymogenic acid, nicotinamine, and some other chelators which are not present in animal systems.
Okay, thank you, Kurabashir. Again, join me to thank him. Now let me invite Mr. Muzammal Haq from Niigata University, and he's going to talk in QTL mapping for stem swelling trait. Please.
Assalamu alaikum. I am just presenting QTL mapping. It is very simple, but from around the last 30 years, from 1990s, very efficient tool for identification of gene, especially for quantitative trait in plants. So, QTL mapping. QTL maybe for molecular biology, we know quantitative trait loci, we know all about that. And kohlrabi, I think Indian region it is very popular and some European region, but not common in Japan or some other countries. So I am just giving information about QTL mapping in kohlrabi for stem swelling trait. Kohlrabi, we know it's a word from German words, and the kohlrabi is a brassica crop. It's vegetable crop, and it originated in 16th century in Europe, used as a food and also animal feed. And it's very healthy food for our body because it contains high amount of fiber, vitamin and minerals. And it also contains some herbal components which reduce or protects the stone formation in our kidney. So it is important, and as a new food for Japan region, I show some recipes. That is in Indian region it is popular, this curry and sharks, and most of them are salads. It also used as a fried, like a French fry, and boiled, pickles, and also soups. So, and stem swelling kohlrabi is important because kohlrabi we eat stem. So, on this basis, for kohlrabi it is important. Another important, some other crops like potato and some other crops have stem swelling pattern, especially in vegetables. So, we think that the mechanism of stem swelling is more or less same. So we are our target to find out the mechanism of stem swelling in plants. And for this reason, we make our decision to find out the QTLs and lastly genes to find out the QTL for stem swelling. This is methodology, as I simply know. First, we chose for QTL mapping generally to related species and most preferably haploid species. And in case here we found one haploid, but for kohlrabi we did not found haploid because it is not so common. So we used GCP04. GCP04 is broccoli and kohlrabi commercial. Then we made cross, and we found a good phenotypic segregation for stem swelling. Some found like kohlrabi, some found like just inverse, and we categorize them into their gradings. And this is the way of our target population development by crossing. First F1, then BC1. We did BC1 because all F1 produce like GC type and no segregation. So we went to BC1 population. And we collected DNA by CTAB method and normal standard PCR profiling. And genotyping, after genotyping, for linkage map, we used AntMap, it is Japanese software, very popular for Japanese researcher. And QTL, it is mostly used QTL